or maybe last week, I mentioned that Chapter 12 was going to discuss predominantly skeletal muscle, a moderate amount of smooth muscle, and a minimal amount of cardiac muscle, because in Chapter 13 we'll better address the cardiac muscle. To remind you where smooth muscle is found, you'll find it in what's considered to be hollow organs. The notable exception is the heart, okay, because we have cardiac muscle in the heart. So organs like your stomach, your bladder, respiratory, lining the respiratory system, the female uterus, blood vessels, etc., is where we will find smooth muscle. And the whole point is to move something through that organ. Okay? So around the respiratory system, it's going to regulate airflow. In the digestive system, it's going to remove food and digested products. Uterus, right? Babies, right? Etc., etc. And if we look at the smooth muscle, it does not have that apparent striation like we saw in the skeletal muscle, and then we will see again in the cardiac muscle. And as well, we are familiar that it's under autonomic control or involuntary. Intracellularly, the sarcoplasmic is present but not as extensive when compared to the skeletal muscle. There are no T-tubules, so we changed up our structure. We just changed up our function. Very good. Calcium is still important for smooth muscle contraction, but it's going to come from a different primary location, and it's going to have a different mode of action. So it's still important, but it's changed up the game. Calcium is sequestered not predominantly in the SR, but actually where? Extracellular fluid. Okay. So our gates, our voltage-gated calcium channels, are mostly in the smooth muscle membrane versus on the SR. They're still there, but not as numerous in comparison. And so therefore, we have our voltage-gated channels, right, to allow for smooth muscle contraction. There are no sarcomeres, but there are contractile proteins. They're just arranged differently. So no sarcomeres, no striations, making the tissue smooth in description and in looking at it with a microscope. Smooth muscles can undergo hypertrophy. They can also undergo a process called hyperplasia. Hyper means excessive plasia. Do you know that one? Division. Okay. So our smooth muscles numbers can divide or can increase. Can our skeletal muscle cells increase in numbers after a certain point of development? No. Can our smooth muscles? Yes. Okay. And the process is called hyperplasia. This is illustrated during puberty at the smooth muscle tissue in the uterus. The abbreviation E subscript 2 is estrogen, or a form of estrogen. The estrogen targets the smooth muscle cells to divide and increase the smooth muscle content. Okay? As well as the same thing during pregnancy. The smooth muscles numbers can increase in that case as well. So smooth muscles can increase numbers, and then obviously they would increase size as well. So they go hyperplasia and what? Hyperplasia, once they divide, they need to grow. So hyperplasia and hypertrophy. Very good. This is a difficult one to get through. Okay. I want to spend a moment on this image. It is very similar to the image that you have out on your handout number 5 that you're submitting today. So you can go ahead and look at either of those, synonymous with one another. In the image in your guided notes on page 7, the cell types are left and right, and in the handout they're stacked, top and bottom, between the different types of smooth muscle. This image will actually kind of unfold the rest of the lecture, so we'll go through it in the image like we've done before, and then we'll formally fill in our handouts. Okay? And so where we're going is we're learning that although there are many types of smooth muscle in our body, they're not all arranged the same. And they're arranged differently, and therefore they have different functions. They work differently. In this image in your guided notes, the gray things are representing smooth muscle cells. So we have 3 smooth muscles on the left panel and 3 smooth muscles on the right panel. Okay? not good. I'm not taking it this time. He's fine. So we have 3 different types of cells represented here. You actually kind of had to look ahead, so you became familiar with what's called single unit versus multi-unit smooth muscle cells. Okay. Let's just look at this image for a moment. I want you to scrutinize it, and you've had to think ahead with homework 5. So I want you to scrutinize it even more. On the far right, we have smooth muscle cells that are considered to be single unit. And on the far left, those that are considered to be multi-unit smooth muscles. So I'll put the recording on pause for a moment. I want you to look at it, review what you have in your handout number 5. Just scrutinize it. Okay, what are you guys seeing? What do you mean? We actually have both types. The one on the top in your handout is what type of tissue? Single unit. The one on the bottom is just and it's labeled as such. That was difficult, right? All right, what else besides the obvious can you see? Gap junctions. What is a gap junction, and or what does it effectively do? Ions distribute through it. So cytoplasm to cytoplasm. We now know that does what? Why? What does that do? Good job. I was getting worried for a moment. Action potential conduction. We can directly pass an action potential from one cell to another to another. So they're effectively electrically connected with one another. What else? What else can you see about single unit cells? Can the autonomic nervous system control them? Looking at your handout, you got your notes? I'll help you out. This says receptor for... So probably which nervous system can control it? The autonomic nervous system does control single unit. Does it control multi-unit? Yes. Okay. So both are under control of the autonomic nervous system. Can they be tropically regulated? Or I guess hormonally regulated? Yeah, sure. They're cells. They better be able to be responsive to hormones like insulin, growth hormone, thyroxine, whatever. So it makes sense that our nervous system can control them, our hormones can control them, blood factors can control them. What else can control single unit muscle cells? Looking at your handout that you're going to turn in a little bit. Something called a pacemaker. We've seen that word before in which organ? The heart. But it's also in smooth muscle cells, okay, that are called a single unit. So in your guided notes on this image, I want you to label this first cell in the single unit section, just label that the pacemaker cell. We're labeling this cell right here in my color in yellow, the pacemaker cell. It sets the what? Initiates the action potential and the frequency of action potentials. Okay. All by its little what? Self. Okay. So single unit smooth muscle cells we see can be extrinsically controlled by the autonomic nervous system. It can be extrinsically controlled by the endocrine system, but it can also be what? What else can happen? What other kind of control? Not much left, option-wise. We said extrinsically controlled by the nervous system. We said extrinsically controlled by the endocrine system. It can also be controlled, intrinsically controlled. Very good. By cells that are called, we just label these as pacemakers. So smooth muscle cells can initiate, some single unit smooth muscle cells can initiate their own contractility by themselves without the nervous system telling them to do it, without the endocrine system telling them to do it, right? They can initiate it themselves. If the pacemaker generates an action potential, it's guaranteed, pretty well guaranteed to conduct through the rest of the cells. From your handout, if I label this pacemaker, what should I label the other cells as then? Contractile cells. So label those as such. Label each of the remaining two contractile cells. The job of the contractile cells is to contract. The job of the pacemaker cells, initiate action potentials. Kind of like a what? Kind of like, really, not so much a muscle, but more like a nerve. Even though they're a muscle cell, they don't really contract, but they do generate action potentials. Before I move on, I want to address any questions that you may have about the role of a pacemaker cell in our single unit smooth muscle versus the contractile cells of a single unit. Let's contrast single unit smooth muscles to those. Actually, before we move on, why do you think the word single unit is used to describe these tissues? Because it's all connected, and all of these hundreds or thousands of cells work together as a single one unit. Very good. Okay. Not all of our smooth muscles are connected functionally, anatomically nor functionally together. Those other remaining smooth muscle cell types we would call what? Multi-unit smooth muscle cells. Are they intrinsically regulated? They are not intrinsically regulated, so they are strictly reliant upon what type of control? Extrinsic control from the autonomic nervous system and the blood factors, whether it be other humoral stimuli or hormones. So the multi-unit smooth muscle cells are completely extrinsically regulated very precisely with the nervous system and as well as humorally. Structurally, what else are we seeing? They have a lot more receptors, which makes sense because they're more reliant upon external or extrinsic control. What else are you seeing or maybe not seeing in multi-unit? No gap junctions. So these cells are all what? Separate units. Okay. Or describe multi-units. They are structurally, functionally independent. They do their own thing, depending on whatever the external stimuli tells them to do. All of the muscle cells that are multi-unit, we can call what? They are all individually what? what do? Their job is to, what is their job? Contract. All of the cells in multi-unit, there are no pacemakers, so they're all individually contractile. So if you want to put in parentheses, they contract their muscles. Is it coming together? Or maybe I should better ask this, what questions do you have about putting this together? Do it. It's slowly coming together. Are these handouts helpful? They're working. Something I should use in the future, you think? Good. Thank you. All right. Now we're just going to fill in the notes. It's going to be redundant from what we learned in the handout and from what we learned in this image. There may be something hiding under, there may be text under that image that got covered up in your handout. So if it is, well, we might have, I can't remember if it was this image or another image coming up, so we'll just see. I think so. Yeah, I think the contractile bit. Yeah. The contractile cells are covered up in this image, so you got to squeeze it in the bottom. And the nomenclature on the bullets also didn't translate. So we learned that some cells are either one, pacemakers, whose job is to be self-excitatory, or you could say intrinsically regulated, right? Okay with that word? Yes, they're self-excitatory or they're intrinsically regulated. But they're also controlled how? Not only intrinsically, but also extrinsically by the endocrine system and autonomic nervous system. Okay. So some of our smooth muscle cells are pacemaker cells that do set the contractile rhythm for a bunch of cells that are grouped together, or this term here is described as sheets. And they're self-excitatory. They spontaneously depolarize without external stimulus, or we said they're intrinsically regulated. The high proportion, like 99% of the remaining smooth muscle cells are contractile. That's why we have muscle cells is there to contract. A minimal amount are there to excite, but a high proportion are there to contract. Okay? And it's the contractile cells that generate tension contract that we're going to describe. Are you okay on the difference between a pacemaker versus a contractile cell? We'll repeat the terminology in the heart. If we look at smooth muscle cell, anatomically it's different in that our smooth muscle cells don't extend the entire length of the smooth muscle organ. So for example, you won't see like a smooth muscle originate at the back of your throat and end all the way down there, right? There's lots of different cells. They're short and branched. They do contract, and they do have myosin and actin, but it's built very much differently. And I've done some research to try to better understand this. Your book kind of plays out on you, because really the research is coming. It's coming now. It's kind of actively being discovered. And so up till now, it's been kind of vague about the real structure of smooth muscle. But there definitely are familiar contractile proteins, myosin and actin, that they're built differently. But their job is to contract. Okay? The myosin in your smooth muscle is longer than that you find in the skeletal muscle. Actin has a tropomyosin blocking molecule. It does not have troponin, though, and I think this is another thing that your book just flat out just says wrong. And it drives me crazy. I've been noticing it the last couple of semesters, so it's flat out. It says that your book says it has troponin and not tropomyosin. Somebody have their book out. Let's go ahead and turn to that, and while you look that up, I'll put this on pause. All right. So just to recap what I just mentioned is that the smooth muscle does have contractile proteins. It does have a thick filament. It does have a thin filament, myosin and actin. Actin has tropomyosin. It does not have troponin. It has a similar kind of role or a protein in there, but it works differently. And there's also some intermediate filaments in there, or cytoskeletal components to help maintain structure. But as I noted here, they don't give contractile ability. They're just giving support to the cell. The way that the thin and thick filaments are arranged in a diagonal pattern instead of like a parallel type of relationship. And so instead of the muscle just completely just shortening end to end like we did with our hand exercise yesterday, they kind of like, as described here, as corkscrew, they kind of twist and shorten. And causes the cell to kind of splay, kind of cool looking how the skeletal muscle, or excuse me, smooth muscle contracts. It's not just shortened, but it twists and convolutes, kind of looking type of structure. So we have our intermediate filaments attached to these things called dense bodies, which in this image are the black circles. And then the intermediate filaments are the blue structures. They're giving support to the thin and thick filaments, which are not illustrated in this image. The role of the dense bodies is similar, right, to the Z-disc in that they give attachment for our thin filaments. Okay. If we contrast the image, right, contracted versus relaxed, you can kind of see how it not only just shortens but broadens. It displays out. Figure 1233 in your textbook illustrates the same thing, but it does a little bit better job illustrating how the thin and thick filaments are arranged much differently in the smooth muscle versus the skeletal muscle. I'd like to back this up with what I was talking about with a fairly recent article, 2010, looking at a smooth muscle. So we can see what's described as a sheet of smooth muscle cells all grouped together. And if we zoom in at a given muscle cell, we can see the dense bodies in the little red things, the intermediate filaments, and the blue lines, and then our contractile units attached to those in the middle. So if we zoom in on our contractile elements, it looks similar where we have our actin thin filaments and our myosin thick filaments. Myosin still has a cross bridge, but it's a different type of myosin, and so it gets excited differently. Okay? So we have a whole, like, almost like a second messenger pathway, this and that and that. And we're going to go there in just a moment. But if we zoom in at the myosin, it's what's described here is the neck region of the myosin that's going to get phosphorylated to allow for its contractile activity. Okay. Where we're headed, can you see this little pink blob in panel B? You all see that? It is called calmodulin. Modulates, regulates calcium. And so what we'll see is, although calcium is important for contraction, calcium does not directly bind to the thin filament. It doesn't bind to the thin filament at all in smooth muscle. It binds to a molecule called calmodulin. And so as we identified, because there is no troponin, calcium is still important, but it binds somewhere else. We still have to phosphorylate our cross bridges. And calcium, we learned, comes from the extracellular fluid initially. That voltage change, though, triggers the sarcoplasmic reticulum to purge out any stores that it has. But the key thing is our first source is extracellular. Then our intracellular sources release its calcium out in the cytosol. What calcium does is then binds to this protein called what? Calmodulin. And it's like a second messenger pathway. Calcium binds to calmodulin. Calmodulin activates something called myosin kinase. You know this now. ASE tells us this is an enzyme that kinases. We already used it. I'll go to my go-to girl. Kinases. No, you used it accurately a moment ago. Well, I said. All right, I'll give you a pass today. Kinases do what? They what? Phosphorylate. Good job. Thank you. Thanks for covering for it. All right. It's an enzyme that phosphorylates myosin. Okay? And so calcium flux initiates a reaction to phosphorylate myosin. It just transfers substrate level phosphorylates the myosin, right, cross bridge so that it can power. All right. Same thing, figure 1234 kind of goes through the sequential steps in good detail. If you look at the image, we can see calcium fluxing from the extracellular fluid through our gated channels. Okay. That voltage change due to the influx of calcium causes the SR to release its calcium. Now remembering, this initial influx of calcium may be due to neural stimulus, hormonal stimulus, pacemaker cell, whatever tells it to do it. Calcium is going to come in from the extracellular fluid and then cause the SR to release its calcium. Calcium binds to calmodulin, and now it's just described calcium calmodulin complex. Calmodulin activates something called myosin kinase, which you know well now. Phosphorylates. Very good. Your textbook goes into the anatomy of this a little bit better. It calls it myosin light chain kinase. If you can handle that, it just phosphorylates myosin, I'm really very happy. Okay? And so that's what it does. It activates the myosin cross bridge and allows for contraction of the smooth muscle. If the smooth muscle needs to relax, what should happen? Calcium's got to get out of that cytoplasm. Back into extracellular fluid and some into the sarcoplasmic reticulum. Put it back where it came from. Okay. Here's another image, fairly recent as well, 2010. 3 panels. We're looking at the membrane of a smooth muscle cell. And this is the cytoplasm in the white area, and the sarcoplasmic reticulum. And you can see the calcium stores in little spheres. What it's illustrating is first extracellular calcium causes the release of sarcoplasmic calcium. Okay? And then, same thing, we see it out. And then if we look in the third panel, you can see calcium binding to calmodulin, is what this is illustrating, that kind of purple complex. And it's the kinase that phosphorylates the myosin. So it's this crazy little second messenger pathway, basically. It's not a direct effect like we saw in the skeletal muscle. Yesterday, the skeletal muscle may have seemed really confusing, but in perspective, it's a little bit more simple when compared to smooth muscle. As well, another 2008 article illustrating the relaxed versus the contracted state of a smooth muscle. You can see the overlapping of the thin and thick filaments in the contracted state versus the relaxed state in the panel above. Next thing you already identified as well is the calcium's got to go back out of the extracellular fluid back to the SR. And that myosin actually has to be dephosphorylated to let go, which maybe makes sense to cause relaxation. Questions at this point? Okay. All right, so if we look at smooth muscle, it can contract, it can generate tension, but it's built differently than skeletal muscle, so it functions differently. So it contracts. However, it contracts more slowly. It contracts like the skeletal muscle. Skeletal muscle was quick and strong, but did what? Quick, powerful, but the offset was it tired out. Used a lot of energy and it tired out. Almost the complete opposite in smooth muscle. Smooth muscle is way slower because we have that second messenger pathway to allow for contraction. It requires less ATP when contrasted to skeletal muscle. It doesn't have to generate as powerful of contractions as regularly. And it also has aerobic respiration as a preferred route of ATP synthesis. So it can make as low requirements and it makes it efficiently. And so not as expensive to run, if you will, when compared to skeletal muscle. Something else interesting about smooth muscle is that it's responsive to stretching it. So think about your stomach or your bladder. Let's think about a big meal. You take in all that food, smooth muscle stretches. That right there causes an intrinsic activity of some smooth muscle to start contracting. Okay, so it does have a stretch mechanism, or like if you drink a big root, 60, whatever they are, right? Taking all that liquid, bladder's going to stretch out at some point. It's going to start. You're going to know that those smooth muscle cells are contracting and it's time to go, right? And so our smooth muscles can respond to stretch. It's kind of like an intrinsic stimulus. Okay. Once we stretch a smooth muscle, it can also maintain that resting length, what's called a stretch relaxation response. Think about a woman who is 42 weeks pregnant with triplets. Okay, so she's like 3 feet out there, right? Okay. Yeah, she's got bigger issues than worried about smooth muscle contraction, right? That smooth muscle, it's terribly stretched, right? Lining the uterus. Okay. But once it stretches out, it still retains the ability to contract, okay, and contract forcefully and well, okay. So at the time, right, when it hits, right, those smooth muscles can contract, right, very powerfully. A more real example is if you eat, right. It's summertime. You probably want to go to a pool right now. Okay. Let's say you're lucky, you're done with lecture now, and you go to IHOP and you eat all you can eat pancakes, and then it's noon, so you got to go to the pool. Okay, so your belly's full of pancakes, and you're going to the pool and you're swimming, right? And the stuff is in your food, or your food is in your stomach. Stomach is stretched out. Okay. But your body now needs to power your skeletal muscles. Okay. So the smooth muscles are on hold, if you will, so they can stretch and accommodate. Okay. Once you're done swimming, then what can the muscles do in the smooth muscle? Okay. Then they can contract. Okay. So our smooth muscle can stretch, relax, but still do its work normally. Okay. And the stretching is way more profound in smooth muscle than the skeletal muscle. You have a limit, a physical limit to how much your skeletal muscles can stretch. Like your arms, right? There's only so much. Your smooth muscle proportionately can stretch crazy. So think about your stomach again, or the uterus, or whatever the case may be. What I want to do is we'll conclude here. I want you to pass your handout 5 to the center of the room. I'll pick those up. If you are late, you need to pick up handout number 6. And we'll see you guys in lab or in class.